All right, guys, today we're going to talk about DNA, its structure, as well as touch on DNA replication. So just to touch a little bit on the history of DNA, DNA was first discovered only in 1953, so we've really only even known the structure of DNA for the last about 70 years now. If we're looking, the first person who kind of started this whole discovery of DNA was Frederick Griffith, and he discovered that a factor inside of diseased bacteria could actually transform harmless bacteria into deadly bacteria. And it was later proven that that factor that transformed this harmless bacteria into deadly bacteria was DNA. There were some other studies as well that we've kind of skipped over this year that kind of led to this point, but in 1952, Rosalind Franklin, who was also working on trying to discover the structure of DNA and what it looked like, she actually produced the first X-ray photo of DNA. And that was really significant at the time because nobody had known what DNA actually looked like. Basically, the image, the X-ray photo, produced kind of like a shadow of it and allowed scientists to figure out what the structure would look like. Now, Watson and Crick actually kind of deceivingly snuck into her office and took a look at this X-ray photo, and using this information, they actually figured out what the structure of DNA looked like. It's actually a really fascinating story. Unfortunately, we don't have a lot of time to talk about it this year. Typically, I show a video, actually, which kind of highlights that. So if you're interested, definitely take a look. DNA controls all of the functions that occur in a cell. But how? Well, it does this by producing proteins. Now, proteins make up a large portion of the structure of our bodies. They make up our muscle, our skin, our bones, our hair, the antibodies, just countless things, infrastructure in the cells, everything. So there are so many things that are controlled by these proteins. And by having the instructions and the code to make those proteins, DNA is ultimately the factors that control everything that happen in our bodies. Proteins are also used to make enzymes, which help speed up chemical reactions. So these enzymes are absolutely essential for our bodies and our cells in doing everything that they can. So what are the components of DNA? Well, DNA is the polymer of DNA. DNA is a nucleotide. If you remember earlier this year, we talked about the different organic molecules. We mentioned that we had carbohydrates, lipids, proteins, and the last one, nucleic acids. Well, nucleic acids, we said the example was DNA. And nucleotides are the monomers of nucleic acids. And what makes up one nucleotide is the following things. The first is a phosphate group. The second, a 5-carbon sugar called deoxyribose, in this case with DNA. And the third thing is a nitrogenous base. And we have 4 of them. The first is guanine, cytosine, adenine, and lastly thymine. And you guys can remember them with their letters, G, C, A, and T. You're going to see them used so much. And how they base pair with each other is vital to how DNA not only operates but also replicates as well. You will need to know the names of these, so definitely study these 4 and also study which ones pair together. Now, if we wanted to see what the structure of DNA actually looks like, well, here's kind of what it looks like. So this is one nucleotide. So again, just kind of like this would be like looking at one Lego piece in the entire castle that you built. Well, if you look, the base here is going to be, actually, it's a little askew on this one, but it's actually usually towards the middle of where the DNA is. The ribose sugar and the phosphate groups that are bonded together, those are going to be on either end. And you'll understand what I'm saying on some future slides. Now, we do separate these based on structure, and their structure actually plays a really important role in how DNA is kind of made, and these base pairings are made. But 2 of these bases, adenine and guanine, are known as purines. I remember it by knowing as pure, P-U-R, as gold, A-G. And these have 2 rings in their structure. And you can see here below in the diagram that you can see both of these diagrams for adenine and guanine below have 2 carbon rings there. Well, pyrimidines, they have only 1 carbon ring. And cytosine and thymine are examples of pyrimidines. Now, if you look, uracil is also listed here. Uracil is a new base, but it's not found in DNA, so we won't be talking about it at all today. We will, however, be talking about it next week when we talk about RNA and we talk about protein synthesis, because uracil is going to play a big role in making these proteins. The last thing to know about purines and pyrimidines, guys, is that A is always going to pair with T, and G is always going to pair with C. And that's the case because one purine always pairs with one pyrimidine. We'll talk more about those base pairings next slide. So looking, here's a diagram of DNA. You can see here that the phosphate groups and the sugars are bonded together with covalent bonds. You might have learned that earlier this year. Covalent bonds are when two molecules are sharing their electrons. And the sugar and the phosphate, they kind of alternate from one another. Phosphate, then sugar, then phosphate, then sugar, phosphate, sugar. And that makes up what we consider the backbone of DNA. If we untwisted DNA to make it a ladder, basically, because that's kind of what DNA looks like. It's called a double helix, but it looks like a twisted ladder. Well, the sugar and the phosphate would make up the sides of the ladder. The bases, on the other hand, thymine, adenine, cytosine, and guanine, those would make up the middle or the steps of the ladder. And that will be a little bit easier. This, remember, is only one strand. So on the other side would be another strand, which we'll see in future slides. Here's another diagram kind of explaining the same thing. You can see here on the top that we have the twisted double helix. That's what DNA normally looks like. Underneath is basically denatured. It's been broken down and split apart. And you can see that those colorful teeth that are sticking out, those are the bases, while the edges, the spiral, that makes up the sugar and the phosphate. So Erwin Chargaff was another scientist. He had studied DNA as well and realized that the amount of A always equaled the amount of T, and that the amount of C always equaled the amount of G. And between the two of those things, he realized that A must always pair with T, and C must always pair with G, because their amounts are equal to each other. Now, Watson and Crick, they actually went out to dinner with Chargaff recently before they made their discovery of the structure of DNA. And after learning this information, they were able to properly structure their model together, basically by joining the two strands of DNA in this double helix. And everything just kind of fit together. Again, a fascinating story. The documentary is called The Secret Life of DNA, if you ever want to check it out. These bases together, the two strands of DNA, are connected by a different type of bond, not covalent bond, but hydrogen bonds, which are much weaker than covalent bonds. You might remember hydrogen bonds when we talked about cohesion and adhesion with water, because water is a polar molecule. Here's another diagram of DNA, and you can see here these solid lines on the outside of connecting these bases with the sugars. There should be some more lines here as well. Those would represent the covalent bonds. The dotted lines in the middle, that's the one I just wanted to focus on. Those represent the hydrogen bonds here, those bonds holding these two strands together. DNA is antiparallel, and what that means is that the two strands run in opposite directions of one another. So as one strand runs up from the 3' end to the 5' end, the other strand runs in the opposite direction. Again, moving from 3 to 5' end, but the difference is that strand is basically laid down upside down. Now, as far as this course, it's really not going to matter too much why, so you don't have to get too caught up in that. It does have a major role in how DNA is replicated, though. And if you look, the 5' end is always the end that has the phosphate at the end of it. And you can see this phosphate on the 5' end of this diagram there at the top, while the 3' end ends in a sugar. So while one has phosphate on the top, the other strand on the opposite side, which it's paired to, has the sugar on the top. And again, that difference is going to play a big role in how the DNA is replicated. This is probably the best diagram out of the diagrams that I've shown you guys so far. So feel free to come back to this slide and take a look. You can see here that we have the phosphate, the sugar, and the base of each nucleotide. And you can see that we have the 5' end on one strand, with the other strand starting at the 3' end. And you can see the oxygen is actually upside down. It's on the bottom on that 3' end strand. And you can see how it's laid down in the opposite direction pretty clearly. So we've already said that cytosine always pairs with guanine and adenine always pairs with thymine. And it's basically the sequence of these letters, these bases, that are going to determine what the DNA is going to do, how the proteins are going to be built, and it's also how we are similar as species with other types of organisms. When we look at ourselves compared to chimpanzees, for example, we share over 98% of the exact same genetic code, the same letters, those C's, G's, A's, and T's. The order of those letters are going to be exactly the same, 98% similar to chimpanzees. So the more similarities that we see in the genetic code, the more evolutionarily related we are to those organisms. It's also a way that we are able to use when investigating crimes. We're able to identify people based on their DNA in their blood, hair, and semen. And we already mentioned the evolutionary relationships. All right, last thing I'm going to talk about, guys, is replication. You guys won't need to know too much about it, but basically DNA goes through what we call semi-conservative replication. And what that means is that we take one strand of DNA and we are going to break it apart, and in each new strand that we make, each of them is going to have half of the old original DNA that we had in it. So the first step is that, and you guys already learned, that DNA is copied before cell division in the S phase of interphase, back in when we were talking about mitosis. So you know when it's occurring. Well, how it happens is first that an enzyme named helicase is going to untwist and unzip that strand of DNA. Another molecule, an enzyme called DNA polymerase, is going to build two new strands by using the original as a template. So if there's an A on one strand, the DNA polymerase knows that it's going to bond a T nucleotide, a thymine nucleotide, on the opposite side. If there's a guanine nucleotide, then it's going to bond a cytosine nucleotide, and that's because those are the base pairs that we always see these match. And by the way, guys, that's the biggest thing you guys need to remember, is those base pairings. These nucleotides, by the way, they're just kind of floating around, and the DNA polymerase is going to grab them and put them on. I did mention earlier that because DNA is anti-parallel, it is going to basically need to be made a little bit different in fragments. So instead of one of the strands is going to be made just completely in one strand, the other one is going to be it's basically like read backwards. So it's going to need to be pieced together at the end. And another enzyme called ligase is going to piece those fragments, called Okazaki fragments, together to make new DNA. And that's pretty much it. After that, we now have two strands of fully functioning DNA. So we'll talk more about this in the next coming days, and next week we're going to talk about protein synthesis and discuss how our DNA is then used to make proteins. All right, thanks, guys.